Good morning, everyone. I would, first of all, thank the organizers to have the opportunity to share this presentation with you today. This is a work made in collaboration between the Center of Ethology and Applied Biology in Murcia and the Center of Automation and Robotics in Madrid, Spain. So I've structured my presentation in 4 parts: the challenge we face, the project, RFID modeling, and short written story. The challenge we face is keeping fruit production in quality and quantity under limiting conditions by the use of the sustainable use of resources from the nutrients and water that the plant uptakes from the soil, and the synthesis of photosynthesis in the root tissues, the leaves, and the transport to the stem tissues, the flowers, and the fruits. What happens normally under any abiotic or biotic stress experiments is that normally source tissues as leaves are much easier to measure and take high throughput phenotypic data, as we have seen before in the talks, that flowers, which are the precursors of the fruits and also the target of pollinators. 2 years ago, we demonstrated in our work published that bumblebees are able, show the preference for genotypes that are high yielding only under an environmental express that was nutrient deficiency. But for achieving this, we had to visually track hundreds of flowers, which is not an efficient method for phenotyping. So what we proposed is to use a phenotyping platform in which we have adapted a system for tracking insects based on radio frequency. So we have an active component, which is the antenna, which is mobile and follows the plant growth and is around in the floral trusses. And this antenna is continuously emitting electromagnetic fields, waves, and when a tagged individual, as a bumblebee, approaches a plant, so every time a tagged individual, which is individually barcoded, is close to one antenna, it generates an RFID signal that it's automatically detected. So this became a reality just last year under a project called Darwin, which is financed by the European Innovation Council and built on 5 partners across Europe. That includes also private companies as well as public research institutes. This is a picture from the sky. You can see here the phenotyping platform. These are the characteristics. We have 6 greenhouse experimental units, so we can replicate controlled environments. And well, let me show you some details of this structure. This is the RFID antenna, which is really light and adaptable to plants. And this is the tag. I don't know if you could see that. Here attached to the bumblebee's body. A greenhouse sector looks like this, and here I think you will be able to see how this bumblebee can pollinate flight, because they are able to carry big amounts of pollen. So it's a factor we control that they can do like their normal life, although they are tagged. And here also with the fellas in the beehive, there's one here and all other one around here that they are tagged, so they can interact with the rest of the beehive members. So we put a big effort to distinguish the pollination-related information from all the RFID detections we registered. That could be from May to June, more than 1 million. So we developed 4 criteria for filtering this data, which is defining, first of all, what actually a pollination visit is, defining a range of time between signal and signal, to be merged into the same single pollination visits. Also the possibility to merge fragmented visits into a single one, and the filtering and the removing of false visits due to extended inactivity, or because they are made at night, as you can see in this daily cycle. You can see the number of visits and a minor number are performed at night, so we just ignored it. And we ended up, in this example, with 14,000 visits corresponding to 137 tagged bumblebees and 287 plants. We consider 4 RFID-derived parameters, which are, apart from visit number, also the total accumulated time from those visits, also the visit average time, taking into account each individual, because each individual could have a different distribution in their visit times. All these parameters are grouped per genotype, and also the number of different visitors and tags. So what we propose is that as well as inbreeding, we use selection indexes for selecting material. We could build up a bumblebee-based index in which we integrate these parameters, and each parameter would derive from a mixed model. Each parameter was modeled according to the distribution by a generalized linear model approach, and the predictor in all cases will be the factor genotype, and we will consider the number of flowers and the distance to the hive as covariance. Also, well, we saw that the genotype effect was more important than the number of flowers, but we have to take this into account, and also the distance to the hive, because bumblebees are learning over time, and also consider the energy cost versus energy reward while they are foraging. And the greenhouse sector is considered as a random effect factor because, there's variability, and also the beehive is different between greenhouse sectors. Also, we set up the same conditions. So the example I would like to talk about is one evaluation we performed in a population of integration lines, in which the commercial tomato was the receptor genome and a wild ancestor, Solanum bipinellifolium as the donor parent. And as you can see, if you look at this pie plot, which shows the mean Z values of the models of the bumblebees-based parameters versus other more agronomical traits like funnel yield or net photosynthesis, the distribution of the lines can vary. It's different. So what we propose is take into account also those genetic lines that are highlighted by the pollinator and not only are high yield performing. We did it under simulated climate change conditions, which are a very strong reduction in fertigation, as tomato is a higher water consumer. And also 3 degrees, the temperature was 3 degrees higher respect control conditions, the optimal conditions used in tomato cultivation. So what could be interested to have a ranking made by the pollinator? We find that there were, because you could be indirectly selecting all the interesting traits. For instance, for visit number and total visit time, we found a significant correlation not only with yield, which would be expected, but also with harvest index. So they tended to visit more those plants that reallocate resources into reproductive tissues. These are the fruits. And interestingly also, the duration of visits and also the different number of pollinators are also correlated with some photochemical traits. So these are parameters that we currently measure. We currently be taking a lot of measurements every month with a fluorometer, and it's very time consuming. So maybe bumblebees could be doing this work for us, and also with leaf area. Lastly, the number of different visitors were correlated with levels of sugars in general, the soluble solid content in the fruit. Also, all these interpolation lines were genotyped, and the selected ones were back rows to perform fine mapping of the integration regions. So our final objective is to know which genetic regions are more associated to the preference of the pollinator. We are waiting for the results of the flowers, which are the organs with the interacting directly with the pollinator, but we don't have it yet. Surprisingly, I'm not focusing on control conditions in this presentation, but I have to mention that the number of flowers under optimal conditions was when all the plants have the same accessibility to resources is correlated with the number of visits, which is expected. But when you apply a stress like we did, this correlation is reduced and is not significant, so there must be something else going on. And the step 2 is to phenotype also elite lines and breeding lines from a private breeding compound, which are teritype, and doing this crosses between the selected integration lines and also the elite and breeding lines. For phenotype, the pre-commercial hybrids also in the phenotyping platform. So to end with the presentation, I just want to add that we performed this in tomato, but could be extended to any crop somehow dependent on pollinators, and that could be grown at the greenhouse. Just want to thank the rest of my colleagues and the mycologists and the lab head of plant hormones, Francisco, and all to you for your attention. Thank you.